Today, we will start our deliberations on PE 630 coursework. And the first lecture in this is on development of dosage form, where specifically we will be discussing the stages of product development. First, let us understand that how the product selection is made in the industry prior to starting the development of the product. The product selection team is a multifunctional team consisting of business development, regulatory affairs, intellectual property, and other representatives who collects the inputs from all the teams and finalizes the product, targeted, marketed, and the timelines of the project. And it then hands over the project to R&D team for development. The R&D team, which consists of chemical research division, formulation development, analytical development, packaging development, perform technical and commercial feasibility of that project, and propose timelines. So this document goes to all the three teams within the R&D, and they interact amongst themselves before accepting or suggesting some changes into the product selection. Once the project selection has been finalized and a formal notification has been issued to the research and development department, the product development starts. This is carried out in different stages, and they are named as stage 1, pre-formulation stage, stage 2, formulation stage, stage 3, scale up and technology transfer stage, and stage 4, which is the validation or the commercial manufacturing stage. Let us now look at the stage 1 of the development, which is also known as the pre-formulation stage. We will look at the inputs that are required for this stage, the scope of activities in this stage, and the output that ultimately comes out from the different activities. The inputs required for stage 1 include components of the drug product, that is drug substance and excipients, and the innovator product in case of the generic products. In generic products, we are trying to develop a product that is bioequivalent and pharmaceutically equivalent to the innovator product. That is why the innovator product has to be available for the development of the generic product. The scope of the activities in the formulation R&D department: first, a literature search is carried out, a physico-chemical characterization of the API, solid state characterization of the drug substance and excipients, and in the case of generic products, reverse engineering of innovator product is carried out, and evaluation of suitable manufacturing process. In the case of new chemical entities, the pre-formulation activity is more elaborate, while in the case of generic product, most of the information might already be available in the public domain with respect to the pre-formulation properties of the API. Because of that, the scope or the extent of pre-formulation studies is limited for the generic products. After doing the reverse engineering or the pre-formulation and the pre-formulation studies, drug substance excipient compatibility studies are carried out. This is the last activity that is carried out in the formulation R&D in stage 1. Analytical division does the development of analytical method for drug substance and analytical testing of the innovator product in this stage. Similarly, the packaging department will evaluate the container closure system of the innovator. The output of this stage is a pre-formulation report and a development strategy note, which is only an internal document that details out the strategy to be followed for further development. These are the activities in greater detail. In the literature search, various standard resources like the pharmacopoeias, FDA's website for summary basis of approval, electronic databases, and other sources are utilized for performing the literature search and collecting the relevant information. An important part of this is the patent evaluation based on the patents that have been listed in the Orange Guide. The physicochemical characterization of the API and excipients may consist of different activities that we studied in our previous semester in the pre-formulation studies. A unique aspect of the generic product development is the reverse engineering of the innovator product, which involves evaluation of the physical parameters such as tablet shape, tablet color, code for punch embossing, pack sizes, container materials, closure types, etc. And some of the other activities have been listed here. Analytical testing of the innovator product is also performed, and an evaluation of the bioavailability study parameters via the CDER homepage for listing and biostudy parameters. Dissolution profiling via USP monograph and FDA prescribed method are carried out. Thereafter, drug substance excipient compatibility testing is carried out using either DSC or the isothermal stress testing. Isothermal stress testing is the more common method for performing the drug substance excipient compatibility studies. The analytical department, as we mentioned, will perform the assay of the product or the drug substance, stress analysis, degradation, and impurity profiling. Similarly, on the innovator product, assay, degradation, and impurity profiling will be performed. Then the packaging department will perform evaluation of the container closure system, which has been used by the innovator company. And this involves the composition of the material, type of the thermoplastic resin and resin pigments, and other aspects of the primary packaging as well as the secondary packaging containers. The studies obviously will differ from pack to pack depending upon what pack type has been used by the innovator company. The scope and extent of the studies will differ. Now we come to the stage 2 of formulation development. The guiding philosophy for the stage 2 is that in all cases, the product should be designed to meet the patient's needs and the intended product performance. Strategies for product development may vary from company to company and from product to product. A company can choose either an empirical approach or a more systematic approach to product development, or a combination of both these approaches. The activities that are carried out by the formulation R&D in stage 2 involve the product development, process evaluation and optimization using one factor at a time or QBD approach, testing of the optimized batch, dissolution profiling and or in vitro in vivo correlationship where relevant, formulation and process challenge studies, accelerated stability studies in multiple prototypes and packs, so that the final prototype and the final pack can be selected based on these studies. The outcomes from this would be the formulation development report, process optimization report, release of tentative specifications, master formula of the drug product consisting of the payment sheet and the manufacturing instructions, manufacturing formula card, stability studies data. Manufacturing card or manufacturing formula card is a brief document which captures the unit formula, the processing instructions of the product in a concise manner. The analytical team in stage 2 performs the analytical method development and validation for the drug product and the analysis of the drug substance in the packaging matrix. The outcome would be the standard test procedures or protocols and validation reports for the analytical methods. The packaging R&D will evaluate the container closure system and will suggest final packaging material specifications. The relevant part of the regulatory compilation that comes out from this is the section 2.3.p and 3.2.p of the CTD, which is in the ICH M4Q guideline. CTD stands for the compiled or the complete technical dossier, common technical dossier. And then it also leads to the generation of technology transfer dossier in abbreviation, which is called as TTD. The details of the studies that are carried out in stage 2 include prototype development, which includes selection and optimization of excipients, diluents, binder, disintegrants, anti-adherent and lubricants in the case of the oral solid dosage forms, process evaluation and optimization, challenge studies, which are the stress studies on formulation and the process, accelerated stability studies for the assessment of final master formula, and accelerated 1 to 3 months stability studies profile. Analytical testing of the drug product and the packaging matrix. So dissolution in USP medium and other relevant media versus the innovator product to calculate the similarity factor, that is the F2 value. Uniformity of content for low active concentrations. Validation of the analytical package, that is assay, dissolution, content uniformity, completed prior to the process qualification. And then the packaging team will do the evaluation of container closure system consisting of the activities that are listed here. These are the templates of the master formula card, which consists of usually 3 pages. The first one is the label claim. The second page is the unit formula. And the third page consists of the brief manufacturing instructions. These are the templates of the master formula consisting of payment sheet and manufacturing instructions. So this is the payment sheet and this is the manufacturing instructions. Payment sheet is also called as the formulation order. Then we come to the stage 3, which is the scale-up stage of the formulation development and is also called as the technology transfer stage. The formulation R&D team identifies the scalable parameters and generates the scale-up batch of the optimized batch formula. They also perform the accelerated stability studies. The analytical team will monitor and release the stability data to the R&D from the batches that have been put on the stability studies. And the packaging R&D will carry out the machine trials and the tooling development. So the outcome would be the scale-up report, final master formula, final drug substance, excipient and drug product specifications, updating the QBD elements if required, generation of the clinical trial samples or the samples for the bioequivalence studies, stability studies data, and generation of a very important document that is called as the local operation dossier. Local operation dossier is prepared at the manufacturing location and contains all the documents that are necessary for the manufacturing of that product at a commercial level. The detailed activities consist of scale-up batch generation, wherein there is identification of the scalable process parameters. For example, impeller speed and chopper speed in the case of wet granulation. Scale-up batch is prepared in a production machine. Identify the scale-up problems prior to the pivotal demonstration batch, or what we also call as the exhibit batch. Confirm the size of the pivotal and the marketing batch after the process qualification. Review of the final master formula, manufacturing process and control parameters, and designate critical manufacturing steps and specify the sampling and testing parameters. Accelerated stability studies are also performed in this stage, which consist of 1 to 3 month stability profile. And the clinical trial or the bioequivalence studies are performed on the batches that were manufactured under the GMP conditions during the stage 3 of development. Clinical trials are applicable for the NCE based products, while the bioequivalence studies are applicable for the generic product. So different requirements of the bioequivalence study, like the fasted or food effect biostudy, shall be performed on the pivotal lots. Biostudy generally is performed on the highest strength of the product. In case the product belongs to BCS class 1 or class 3, it may be applicable for a biowaiver, and we can conduct in vitro dissolution testing in 3 different pH media on lower dosage forms and perform similarity test on dissolution results to get a biowaiver from the regulatory agencies like US FDA. The last stage of development is the stage 4, which is the commercial manufacturing or the validation stage. And the emphasis here is to establish scientific evidence that the process is capable of consistently delivering a quality product. The formulation R&D generates the validation batches. The packaging team provides the packaging support during the commercial scale manufacturing. The analytical R&D collects and analyzes the stability studies data for regulatory submission and shelf-life extension, and review of the added overages if applicable. The outputs from this stage include process validation report, commercial master batch production and control record, and process flow diagrams. The detailed activities include validation batch generation, where you execute process validation of the consecutive marketing lots, show intra-batch similarity as well as inter-batch similarity between the bio batch and the commercial validation lots. The stability studies on the stage 4 samples are used for the regulatory submission and for shelf life extension and review of the overages. So this is an important stage that for the shelf life determination, samples from stage 4 are utilized. This is the guidance for industry document from US FDA for process validation, and the 3 batches required for validation is no longer valid, and only 1 batch can be taken for the process validation. According to the revised FDA guidance documents, the 3 basic requirements for process validation are: samples must represent the batch under analysis, sampling plan must result in statistical confidence, and the batch must meet its predetermined specifications. Now we come to the developmental timelines that are applicable for different stages. Stage 1 requires 2 to 3 months. Stage 2 requires 4 to 5 months. Stage 3, 3 months. Stage 4 requires 1 to 2 months. And the approval time for the ANDA is 18 to 24 months. Hence, the total timelines in India will be 10 to 13 months, while in US and Europe, because of the extended regulatory approval time, these will be 28 to 37 months. This is the end of the presentation related to stages of formulation development. Thank you very much.